This is podcast 17. We've been talking about getting glucose to the point where it's at pyruvate, that's in glycolysis, and then you import that pyruvate into the matrix of the mitochondrion and decarboxylate it, making acetyl-CoA. You do that with pyruvate dehydrogenase. Then you are going to add that 2-carbon molecule acetyl-CoA to a 4-carbon molecule oxaloacetate and make citrate. And then you're going to rearrange that citrate molecule, and the end result of the citric acid cycle is several reduced electron carriers, NADH and FADH2. A lot of people focus on the ATP that is made in the breakdown of glucose, and ATP is a decent thing to focus on, but these electron carriers, NADH and FADH2, they carry 90% of the potential energy that is taken away from that glucose molecule. And now we're going to talk about, during the electron transport, how you convert that potential energy into ATP. So those high energy electrons are going to be removed from NADH and FADH by oxidizing them. So they were reduced in stage 1, glycolysis, stage 2, the decarboxylation of pyruvate to make acetyl-CoA, and then stage 3, which is the TCA cycle, Krebs cycle, citric acid cycle, all that sort of stuff. That's where NAD and FAD were reduced to make NADH and FADH2. Now you're going to oxidize those molecules in the electron transport chain, and the energy that's released is going to be used to move protons from the matrix into the intermembrane space. So this little space between the outer and inner membrane, you're going to pump protons into that space, and you're going to create a gradient of relatively low pH in the intermembrane space, relatively higher pH in the matrix, such that protons are going to want to flow back into the matrix through this FOF1 complex. Okay, the energy of these electrons is going to be lost gradually as they're passed along the electron transport chain. So energy from NADH is released to an electron carrier, it's passed to another electron carrier, and so on down the chain. These electron carriers are proteins. Most of them are embedded in the intermitochondrial membrane. This is why the intermitochondrial membrane is so membrane rich. It also contains the FOF1 ATPase. A couple of them are soluble. Coenzyme Q and cytochrome C are soluble. We're going to see cytochrome C again when we talk about apoptosis or programmed cell death in cells. It's an important component of that, but it's an electron carrier. So these high energy electrons on this energy potential scale here, high to low, they lose energy gradually as they're passed from one electron carrier to another along these protein complexes that are embedded in the intermitochondrial membrane, or soluble, associating with that intermitochondrial membrane, but can go in and out of that membrane-associated space and also the intermembrane space. That's true for coenzyme Q and cytochrome C. And as they're passed from one electron carrier to another, they lose energy. That energy is used to move protons across the membrane. And there are definite sites that protons are going to move across the membrane into the intermembrane space against their concentration gradient. That takes energy, and the energy comes from these electron carriers losing energy. The final place where the electrons have to go is to oxygen. That is catalyzed by this last complex, cytochrome C oxidase, which is a very large multi-chain enzyme. It's one of the examples I gave you for quaternary structure in a protein, where there are 13 different chains coded for by both mitochondrial genome and the nuclear genome, and they all have to assemble in the inner mitochondrial membrane, otherwise you can't make energy. And then the terminal electron acceptor is oxygen, molecular oxygen. It says O2 here, it's actually O to make O2 with hydrogen to make water, actually. This is why you need oxygen to breathe. You need this terminal electron acceptor. If you don't have this terminal electron acceptor to accept the electrons at the end of electron transport, the whole electron transport chain backs up, and so does everything inside of mitochondria. So no citric acid cycle, no stage 2 acetyl-CoA formation, and you're left with only stage 1, which is glycolysis. Okay, another thing that's important about this is that NADH enters at one spot and passes off its electrons to complex 1, and FADH2 enters at a different spot, enters at complex 2. So complex 1 passes off to coenzyme Q and then complex 3. Complex 2 enters at, or FAD enters at complex 2 and then passes off to coenzyme Q and then complex 3. It's not that complex 1 passes off to complex 2. There are two different entry places to this electron transport chain. Okay. At complex 1, as you're passing off those electrons, right here, complex 1, the electrons are losing energy as they get passed down these electron carriers, and that energy is used to move 4 protons across into the intermembrane space. At complex 2, it's also passing off those electrons from FADH2 off onto coenzyme Q, but no protons are passed off. Complex 3, you get 4 protons moving from one side of the membrane to another into the intermembrane space, and then in complex 4, 2. So again, complex 2 is different. Complex 2 only accepts electrons from FAD. That's the only way FAD enters the electron transport chain. And in the NADH pathway, you go from complex 1 to 3. That's what's depicted here. You see there's no complex 2 on this diagram. So in complex 1, you have NADH being passed off its high energy electrons to electron carriers in complex 1. This is a complex of proteins embedded in the membrane. As those electrons are losing energy, that energy is used to move protons against their concentration gradient into the intermembrane space. Those electrons are passed off to coenzyme Q, which is fluid within the membrane, the inner mitochondrial membrane. Then we reach complex 3, and complex 3 has other electron carriers further stripping the energy from those electrons, and that energy that is stripped off is used to move protons again from the matrix to the intermembrane space against their concentration gradient. Then it's passed off to cytochrome C. Cytochrome C is water-soluble protein electron carrier in the intermembrane space. That passes off electrons to complex 4. Complex 4 is cytochrome oxidase. Cytochrome oxidase is this large multi-chain complex protein that we talked about, and as an example of protein quaternary structure. And ultimately, in complex form cytochrome oxidase, those electrons are going to get passed off to oxygen to make water. Oxygen is a terminal electron acceptor. It grabs some hydrogens from the matrix and makes water. And then hydrogens at the same time are used to move, are moved against their concentration gradient using the energy of electrons that are lost. That energy loss is coming from being passed from one electron carrier to another, and is used to move protons against their concentration gradient into the intermembrane space. Then you end up with a whole bunch more protons in the intermembrane space than in the matrix. This is a proton gradient, so more here than there. They want to fall back down their gradient. They do that through the FO channel, and then the F1 ATPase uses the movement of those protons to physically move and create ATP, and we'll talk about that in more detail. Okay, so this is coming from NADH. Okay, once you make that ATP, you got to get it out of the matrix, and there is an ADP ATP transporter that does that. There are ways that we understand this model from special cases. So this idea of chemiosmotic synthesis, where you're creating a chemical gradient of hydrogen ions from one side of the membrane to another, is a relatively new idea. Not new in terms of your life, but new in terms of science, in that it was Peter Mitchell's idea that he proposed in the 60s about how it is that ATP is made. Most of the ATP is made. And it was not accepted originally that there was this proton gradient that is used to make ATP, and eventually lots of data came to support that, and some of those data came from experiments like this. So DNP stands for dinitrophenol. It is a drug, and this dinitrophenol drug can move protons from one side of the membrane to another side of the membrane. So the electron transport chain is creating a gradient, and DNP, if it's present, this drug is present, is destroying the gradient. So electron transport is still happening, but you're not getting the benefit of the proton gradient. And when you destroy the proton gradient, you destroy your ability to make ATP. So DNP will destroy the ability to, or reduce the ability of mitochondria to make ATP. And it was given foolishly as a diet supplement to help people lose weight. This is back in the 20s and 30s. Some of those people died and had heart problems because they were severely compromised in their ability to make ATP. So if you destroy that gradient, you destroy much of the cell's ability to make ATP, because that gradient is the thing that allows the FO-F1 ATPase to work. And if your F1 FO ATPase doesn't work, then you are stuck with glycolysis as a way to make ATP. It also leads to heat production, and these people had fevers and sweats and that sort of thing. And that is why that is that you are still taking the potential energy from that NADH and FADH molecule. Remember, 90% of the potential energy that is taken out of glucose is stored in those NADH and FADH carriers. And now you're just dissipating that potential energy instead of harnessing it in the bonds of ATP, because you're destroying the gradient. You're making a gradient and destroying it with this drug. Then that energy is just dissipated and not captured in the potential energy of ATP. And when energy is dissipated, it's dissipated as heat. So there's another protein called uncoupling protein, which is natural. It's not a drug. And talk about that in another slide. Okay, so here's an H5P question that needs some background. So the Warburg effect. Otto Warburg was one of the original studiers of glycolysis, and one of the things he noticed was that cancer cells have very high rates of glycolysis, and cancer cells will choose glycolysis even if oxygen is available. So normally we think of glycolysis happening in the presence of oxygen and then continuing on to oxidative phosphorylation, the stuff we're talking about using the mitochondrion. But in the case of cancer cells, even if oxygen is present, they will stop at glycolysis, and they will use glycolysis almost exclusively for energy production. Why do they do that? It seems that it's not an energy sort of situation as it is as much a carbon skeleton sort of situation. By ripping apart glucose, which is usually readily available, you have carbon intermediates that can be used to make other things that fast-growing cells need, like parts of lipids, parts of proteins, parts of nucleic acids. And that seems to be, quote unquote, the motivation for doing it. They have a modified type of glycolysis enzymes that allows them to do glycolysis really, really fast. So normally you would not be able to support a fast-growing cell on just 2 net ATPs per glucose. However, it's sort of like if you are measuring the flow of water through a straw versus through a storm sewer. The storm sewer has a much bigger diameter pipe, and so you think a lot more flow is happening through that storm sewer. But if you put the flow through the straw at very, very high pressure, you can achieve high flow. And so it's by just making glycolysis happen really, really fast in cancer cells, they can get away with it, and they can get those carbon intermediates to build other parts of the cell. That is called the Warburg effect. Okay, now you can answer this question, hopefully. Okay, so I said I was going to mention uncoupling protein. Uncoupling protein is also called thermogenin, or UCP1. Uncoupling protein 1 was one of the first in this gene family to be identified. Now we know that there are many different kinds of uncoupling proteins, 2, 3, 4, 5 at least. And but this is the only one that I've seen where there is really good evidence where it actually uncouples. And the idea is that it's uncoupling electron transport, moving down the electron transport chain from oxidative phosphorylation. So you still have electron transport, but because you don't have the proton gradient, you don't have the proton gradient that is needed to make ATP through the FO-F1 complex. So in the case of DNP, back here, it just binds a proton on one side, diffuses through the membrane, and releases it on the other side, and that's how it destroys the gradient. In the case of uncoupling protein, it's a pore that allows the hydrogen gradient to be dissipated. So this is sort of like having a hole in the bottom of your boat, in that you can continue to bail your boat, but water is always coming in, and you can't. You can try to establish a gradient, but you can't keep that gradient. The gradient keeps on being dissipated. So early on in uncoupling protein research, it was noticed that human babies had uncoupling protein expression in an area of fat um, um, just underneath the skin in their shoulders. But then they stopped making that, stopped expressing that protein after a few months after birth. And uh, newborns are famous for not being able to regulate their body temperature that well, and that was the story. We make this protein in order to regulate your body temperature. It was also seen in some cold climate mammals like reindeer make uncoupling protein. And if you looked at this adipose tissue, it was unusual in that it was brown. Most adipose tissue is white, it has a very low concentration of organelles. This was brown because it had a high concentration of mitochondria in the fat tissue. And the reason it was brown is these electron carriers have cytochromes that have heme in them, which gives them a brownish color. Well, just in the last 10 years, it's been discovered that adult humans also make very small quantities of brown fat. We're talking milligrams in a 200-pound person, and it's expressed in the same places as it is as a baby. Also, some of it is seen in bone marrow, and it generates heat. And there's a lot of interest in this because it is very energy wasting as a tissue, and because there's an obesity epidemic in the adult human population. There are lots of developmental scientists and physiologists looking at how we can turn on these genes in adipose tissue so that we can lose weight. Okay, these are thermal scans, by the way, and just showing that these areas are super warm in this person. And the amount of brown fat that a person would express is very variable among people, and it is one of the reasons why a thin person may be thin, is that they're expressing more brown fat. Okay, so now sorry, Siri's talking to me. Okay, now we have our regular proton gradient. Let's say we're not taking DNP and we are not expressing thermogenin, and we have a high concentration of hydrogen in the intermembrane space and a relatively low concentration in the matrix. It'll flow through the FO part, which is the channel part of the FO-F1 complex, and then as it flows through the FO part, it's going to bind to protein amino acid residues on one of the complexes of that channel. It's going to cause a conformational change. That conformational change is transmitted to a central spindle called the gamma subunit. That will cause that gamma subunit to rotate physically in space, rotate around on its axis. It is shaped like a cam. It's not like a smooth rod, but instead it's got a bump in it. So as it rotates around, it will cause these alpha and beta subunits to change shape. And as they change shape, they're going to literally push together ADP and P to make ATP. So they bind ADP and P, and as they change shape, they push those two molecules together, and that makes ATP. This is a more realistic view of the FO part and the F1 part. If we look at the top-down view, so looking from this surface down, looking at the F1 head, these alpha and beta subunits, there are 3 conformations of the beta subunits. The alpha subunits are in between, but we're not considering those at this point. The beta subunits are the ones that have the ADP and P and ATP binding sites. And then what you're seeing here is this gamma subunit sticking through, and again, it's shaped like a cam, so that as it goes around this way, it's going to bump one of these 3 subunits and cause it to change shape. And it moves 120 degrees at a time, 3 times moving to make a circle, and it will cause conformational change in each one of these as it goes around. So let's start with the O, or open conformation, where ADP and P will diffuse in from the matrix and bind. And then the gamma subunit rotates as protons bind to the C subunit of the FO channel and cause it to change shape. This then causes again, just following the beta 1 subunit, to change shape to a loose conformation where it's loosely holding ADP and P. They're not free to diffuse away, but it's not holding them tightly. One more rotation of the gamma subunit, and it goes to the tight conformation where ADP and P are smushed together to make ATP. And the same thing is happening on these other beta 2 and beta 3 subunits. So that when this returns to the open conformation, ATP will then leave and new ADP and P come in. So we talked about before how ATP is a special situation because you have charge repulsion and that third phosphate is easy to break. Well, how do you make that bond? How would you drag something that is really electronegative next to something else that is really an electronegative and expect to form a bond? If you are trying to do this in solution where ADP and P are just bouncing around each other, it is not very likely to happen in that it is not going to happen spontaneously as a positive delta G instead of a negative delta G. However, in the environment of this beta subunit, where the movement of these molecules is restricted and the orientation of these molecules is dictated by how they're bound to this, the delta G is close to 0. So not exactly spontaneous, but not non-spontaneous, and the action of the beta subunit pushing those two together is enough to make this reaction happen spontaneously. Plus the movement of protons provides the energy for the conformational changes that allows this reaction to happen. So the free energy change in the environment of a beta subunit is not the same thing that when you measure them in free solution. And it's much more favorable on a beta subunit. Okay, let's see. No, I want to show you this movie of a F1 moving. Okay, so here's the FO channel. Here's the F1. Protons are moving through. These are ATP molecules bouncing off. This is now just focusing in on the head, and that blue is the cam. So as it moves around, these subunits change shape, and they go through the 3 different phases of open, loose, tight, So open binds ADP and P, loose holds them, and then tight squeezes them together, and then back to open, ATP gets released. Open, loose, tight. And then back to open, and ATP gets released. Okay, ADP and P that are brought together to form that terminal phosphate bond there. Okay, you get the idea. Okay, so just to review, NADH passes off its electrons to complex 1, and then coenzyme Q, complex 3, cytochrome C, complex 4, off to oxygen, terminal electron acceptor makes water. As it is doing this, protons are moving through these complexes from the matrix into the intermembrane space, creating a gradient, which will cause high protein concentration to go to low protein proton concentration, not protein, proton concentration. They move through the FO complex, bind to that C subunit, which causes conformational change in the gamma subunit, which is the CAM, which is going to rotate around and cause the beta subunits of F1 to go through the three conformations open, loose, tight to make ATP. If you are dumping off your electrons from FADH2, they're going to enter in complex 2 and then coenzyme Q and then 3 and 4. So 1 never passes off to 2, and complex 2 doesn't move any protons. This is a nice little diagram, but it's not like these guys are all lined up next to each other in the cell. All these proteins are moving fluidly in the inner mitochondrial membrane, and they're bouncing in and out from each other. Okay, there are other things that mitochondria do besides make ATP, and those things take advantage of the gradient that is created between the intermembrane space and the matrix. And if the gradient is used to do those other things, okay, like move citrate around, then there is less of a gradient to make ATP. So it means that theoretically it's possible to get a 38 ATPs out of an ATP, but it depends on what else the mitochondrion is doing with its gradient. How I usually describe this is your car mileage. You get, if you got a new car, there's a sticker saying this is how we've measured highway and around town mileage, potential mileage, but your mileage may vary. It all depends on things like how full are your tires, what are the road conditions, how long are you waiting in traffic, how much of an incline, what's the wind. All those things can affect your mileage. Okay, I want you to think about this for H5P. So actually, there is no effect when coenzyme Q is supplement trials are given. The reason for this is probably most people with a decent diet have as much coenzyme Q as you need. There's not a shortage of coenzyme Q in most people, and more is not necessarily better. So although in theory coenzyme Q could be beneficial for exercise capacity and decreasing oxidative stress, in this study and in several others, uh, no difference in supplements. And that's the case for most supplements. Is most supplements show that there's no difference in whatever they're trying to help.